For the ones who don't know me, I'm Martí Català. I'm a senior data scientist working in the Oxford team. Mainly, I work on building R packages, and like here, what I'm going to present is like, well, I will try to stick to the 10 minutes that I've been given. So just complain if I uh, do more. But like I will try to present in these 10 minutes the drug utilization package. That is like, as the name is like indicates, it's like a package, an R package to conduct drug utilization studies. So just a bit of context. What's the drug utilization studies? I hope that more or less everyone is familiarized with it. But if according to the World Health Organization, so it's like a study related to the marketing, distribution, prescription, or and use of medicinal products with like and all their like consequences and so. So basically, in this package, we try to focus in like a bit and in a smaller scope that is basically in Darwin EU has a standard catalog of analytics. So we try to provide the tools to conduct a drug utilization study following this standard catalog of analytics. Also, we try to expand the scope of it in the future, but like this is what we are currently focusing. And so basically, the main functionalities of this package, it's you can create a cohort of drug users, you can compute the daily dose of a given exposure, identify potential indications of like your drug users, summarize the drug use in terms of dosage, quantity, and time exposed. Also, we have like two bits of part is like identify alternative treatments and analyze the treatment discontinuation to see how long individuals are taking these drugs. So basically, currently this package is on version 0.6. It's on CRAN, and we have a nice website. In fact, when I'm going to finish, I'm going to share the link of the slides and the link of the package. But all what I'm going to present, it can be seen like in the vignettes of the package. Well, probably not that one, but like here you can see like more all the documentation of the package. So yeah, just briefly, the first thing that we need to do is to create a CDM object. If for people who are not familiarized, like a CDM object is just a link to all the tables in your database, to all the standard standard tables. If you want to practice, you can like just copy this code that will download the Eunomia database that everyone hope that knows, and it will create like a CDM object. Basically, CDM object is defined by CDM connector, and like it looks like this. Like we have like a link to a set of tables, and like if you do CDM dollar person, you would see the person table. And we, well, you can investigate here in like the CDM connector package. Hope that some of you have already been exposed to it and explains how to do it. Like, and yeah, just like with the Nomi, I had to do some corrections because like it's not the best database to conduct drug utilization studies. But basically, the first thing that we need to identify like drug users, we need like a concept set. This concept set can be defined in many different ways. We can define a JSON concept set. This is like probably the most the one that everyone is familiarized with. But we can also like identify it as an ingredient plus all the descendants with a code generator, or doing like a systematic text search the same way that we would do it in Athena. These are functionalities from Collisionator. Or also like you can define, okay, I have my concept one that is like these concepts 1, 2, 3, 4. So once we have our concept in this example, all the code that I'm going to show, as you can see, like you will be able to copy and paste it, and you will be able to follow like the presentation if you want. So you can create like a code list that is like basically all the descendants of acetaminophen. These is the ones that you get in eunomia, but if you would have like a bigger database, probably you will have way more codes. And once you have your concept set, what you can do is like to create your drug utilization cohort. So basically, what you have to decide, you have to pass like a CDM object and just like put a name and a concept set. If you do that, you would like have as a result like your CDM back, and as you can see, you will have like a new cohort table instantiated. In this case, it will be named DUS cohort, but like the name that you would choose, like it would be here. And you would be able to access it with the dollar symbol at the same way that we accessed it before to the person table. So then like this cohort has some settings that you can check. You can check like the cohort counts with this command. You can check also like the attrition in the sense like this is a very simple cohort. We don't have attrition, but later we are going to see an example with like a bit of an attrition, and you can also summarize the data. So let's look a little bit of where it's hidden. So like now we use it like only the parameters that are compulsory, that is like CDM, the name and the concept set, but we can like also choose like a duration range, input duration, gap era, prior use washout, prior observation, cohort date range and limit. Let's go through them a little bit. So basically cohort date range, it allows us to define an like a study period. Then if we define a gap era, what we are going to see is like all the exposures that are separated by less than this time will be merged into a single one. So these two that overlap and these two that are separated by less than 29 days, they will be merged into a single one. If they are like a bigger separation than gap era, they are considered as two independent exposures. This is like because maybe we consider that if a person stops the treatment for just 2 weeks or 30 days and it starts it again, we consider that like it's within the same episode. So this gap era allows us to define which is the time that the person has to stop. So we consider it's a new episode. We can like also use like a prior use washout, like saying how many days we need to consider it like an incident, new drug, or like to trim the period of that we are interested, if we only want to conduct the study in a certain years of interest, or like if we want just the first one. As you can see, I'm going like a bit fast. I want to show more or less the whole functionality. If someone is interested, I will share all the links at the end, and hopefully they can look at it like slowly. So here, like for example, I do like an example where I say, oh, okay, gap era is 30 days. Any prescription separated by less than 30 days will be merged into a single era. Prior use washout of 1 year, prior observation of at least 1 year. I'm only interested in records between 2000 and 2019, and I just want the first record per person, and then I would get my cohort. As you can see, all the parameters that I used get recorded in the settings. I can get like the cohort counts. As you can see, they are like smaller than before because I applied more restrictions. And the nice thing is that now all my decisions get recorded in the attrition. So here you can see it like in more details, but like we have a plotting function to plot like this nice diagram for attritions. So you would say, okay, initially I had like 14,000 records for nearly 3,000 individuals. When I started joining and like making the different decisions, which was the impact that it had, and we have like the final number of subjects and records. So yeah, so like we can also create the same thing. Now we created with like a custom concept set. You can like specify like for example an ingredient or an ATC cohort set. And yeah, the second bit that I'm going to focus is in add daily dose. Basically, add daily dose, it's a function that relates like your concept ID with an ingredient and via the drug strength, and then computes like the daily dose of each one of your prescriptions in drug exposure. So for example, Here we can see which are like the formulas that are used for the different patterns in the drug strength table. But in daily dose, we can calculate the daily dose of drug exposure. We only have to provide a certain ingredient concept ID, and then we have like an extra column with the daily dose and the unit used. So I see that they've been speaking for nearly 10 minutes. So I'm going to like show the rest of the functionalities very fast in just 1 minute, just to go like give an overall overview. Like once we give like the daily dose, we can like characterize it in characterizing in terms of duration, quantity, and dosage. So we can have like things like which were like the number of records, which was the initial quantity, which was the cumulative quantity, which was like the initial daily dose, the cumulative daily dose, and we can get all of these fixed on like certain parameters and related to an particular ingredient. And then we have some functionalities to show like indication in the prior days where that we can summarize. And finally, we can summarize alternative treatments. As you can see here, like what happened. I have a person, like in all the people that were taking like acetaminophen, my example, what happened in the following month. So most of them were still like in acetaminophen, like in the next 90 days, but like after these 3 months, most of them changed to untreated, but you could have potentially like some patterns here. And finally, and the last, and with this I finish, we have some functions to analyze the discontinuation. So basically what we treat it as a survival analysis. Now Kim is going to speak about it. So hopefully it's a good introduction for the next tutorial. And basically we are going to treat like end of treatment as a survival analysis. So yeah, it was like a very brief tutorial. For the ones who are more interested, we have website, we have like this slides presentation, and like hopefully also we are going to see each in the OHDSI Global Symposium. So thank you very much to all the contributors, and hope that more or less everyone could get an idea of what drug utilization package does.